Hello, hi. Welcome back to the final part of the lecture of inheritance biology, which means the chapter number 4, inheritance or principles of inheritance and variation. Okay, this is the final part and the last part, which means the part 5, I think so. And this is the final part of the lecture. Here we are dealing with the last part of your chapter, which is the mutation. In this lecture, we will be dealing about what is mutation. Okay, and we will deal about certain diseases which are due to this mutation and chromosomal abnormalities. Okay, so coming into the chapter, first we will be discussing about mutation. What is actually mutation? Mutation is the phenomena which results in the change in the DNA sequence, okay, which can consequently which can leads to the change in the genotype and also the phenotype of the organism. This is the thing. What's mutation? Mutation is actually the change in it is a phenomena which results in the change in the DNA sequence, which consequently leads to the change in the phenotype and the genotype of the organism. We know that we human beings or any other organism have cell which contains the nucleus. We have studied that the genetical material is found inside the nucleus in the chromosome, right? We know that chromosome is actually a DNA molecule. The highly condensed DNA is known as the chromosome. So, if there is any change in the DNA sequence, if you need to understand this very clearly, we need to go to the next chapter, in which we deal with the molecular basis of this inheritance. We will deal about the chemical structure, so we can understand it very clearly. So first, now you understand that we have a chromosome. In it we have a DNA. DNA consists of certain molecules. DNA contains certain molecules which are known as nucleotides. Okay, which means the DNA is made by nucleotides. Polynucleotides means many nucleotides arranged will form a DNA molecule, which is actually a genes, right? So first I am drawing just a chromosome here. This is a chromosome. We know that genes are present in here in the DNA. This is actually a DNA. We know that after S phase or synthesis phase, the DNA will be replicated to form two DNA or two chromatids will be formed, right? So I am saying that in a DNA, we know that certain genes are there. DNA contains certain genes, which is the coding information, which contains the genetic information, right? So what are actually genes? Genes are actually the part of a DNA which are certain combination of nucleotides. Now just understand like this, because we will study later what is nucleotides, what is the structure of DNA in the next coming chapter. Okay, now you understand. So in a DNA, if in case of a particular gene consists of certain combination, certain arrangement of the nucleotides. Okay, if there is a change in the nucleotide, the gene will sometimes not express, right? We know that gene need to be expressed. We have studied in genetics, right? Certain characters are formed, the tall height or the dwarf height. Okay, these are caused due to the expression or the not expression of the genes. If gene is expressed, they will produce certain proteins which will leads to the height, which will leads to the tall height of the planet. If the proteins are not expressed, it will not form much height. We have studied in polygenic inheritance also, right? Polygenic inheritance, the genes can cause an additive or a cumulative effect, in which the each genes produce certain an equal amount of effect. So if there is a mutation in that gene, the gene will not be expressed. So what is mutation? Mutation is the phenomena which mutation results in the change in the DNA sequence. So there are some factors, there are some reasons for the DNA to be mutated. Okay, there are certain factors for the DNA to be mutated. We will study the factors. Factors may be due to spontaneous. Mutation may be of spontaneous. We can classify mutation as spontaneous and also mutation which are induced. Okay, we can induce mutation. We will study it right now. So I think you have understood what is the mutation. I will say the definition once more. Please write it down. Mutation is the phenomena which results in the change in the DNA sequence, which consequently results in the change in the genotype and the phenotype of the organs. We know that if the genotype can change, phenotype will automatically be changed, right? That is the case of mutation. We will study, if you need to understand it very deeply, we will be doing it in the next coming chapter, because we will study the chemical structures, so you can understand it very clearly. Okay, right. So that is the case of mutation. We know that apart from recombination, what is recombination? I have said it in the last lecture. Apart from recombination, mutation also can cause the genetic variation. What? Apart from recombination, mutation is also the reason for the genetic variation in organism. I have said in the last lecture that recombination can also enhance the genetic variation. Sometimes in a cell, if there is a crossing over, the genotype can be different from that of the parental genotype. It is a recombinant or non-parental. So there is a change from that of the parent. So there is a change in the next generation, which is the source of genetic variation. Okay, this is the case of mutation. So mutation is the source for genetic variation. Okay, and next we are going to study about point mutation. What is actually point mutation? Point mutation is a mutation in the specific site of a DNA, which means if I am taking a DNA here, taking a double-stranded DNA, we will clearly understand what is double-stranded DNA. I will clearly say in the next part. Now just consider it as a DNA. Okay, a double-stranded DNA means there is a one strand here and there is a other strand here. It is actually they will be having certain nucleotides. I said that DNA is actually a polynucleotide. Polynucleotide means many nucleotides arranged in a particular sequence. One nucleotide here, one other nucleotide here, like. Okay, we have adenine, guanine, thymine, uracil, cytosine. Okay, these are the basic nitrogen bases in the nucleotides. We will clearly say in the next chapter. No need to be worried. Okay, now please understand this. In a double-stranded DNA, what is double-stranded DNA? DNA is usually double-stranded in nature, which means a DNA will be like this. It have one strand, also it has the another strand. So consider it as a gene. We know that it is a gene, right? So it will be having certain sequence here. I am just writing a adenine, thymine, uracil. Sorry, uracil is seen in RNA. Adenine, thymine, cytosine, okay, guanine. Okay, these are the things. Adenine, thymine, cytosine, guanine, any of the order. We can't say which order will be the gene, because it is a random. We can't predict. Okay, because each gene will be having its own combination of the nucleotides. So if this, consider it is a gene for height, if this express, the organism will be tall, because dominant is the character. Dominant is the tall form for the height. Okay, so if there is a, we are dealing with the point mutation. If there is a mutation in a specific site or a specific nitrogen base, then it is known as point mutation. A mutation which occurs in the specific site of the DNA is known as point mutation. Okay, these are the basic idea of mutation. We will deal it very clearly in the coming lectures. So I earlier mentioned that mutation can be spontaneous in nature, and also mutation can be induced. Spontaneous nature means the mutation can be caused due to the internal activity of the cell. Okay, and also the mutation can be also induced by us or any other phenomena. Some of the inducible mutations are from the origin, like we can induce it by certain radiation or certain chemicals or certain fungal toxins. They are chemical in nature. For example, some of the mutating agents are radiation. Radiation are actually waves, right? So we know that waves or the radiation can be divided into ionizable radiation and also non-ionizable radiation. Some of the non-ionizable radiation are UV light. Okay, UV light are non-ionizing radiation, and certain ionizing radiation are alpha, gamma rays, X-rays, these are certain ionizable radiation, which means they can ionize a molecule. Non-ionizing radiation means they can't ionize a molecule. So, consider a human being hit with the UV light. We know that if we go outside, we are prone to sunlight, right? Sunlight can fall into our body. So, sunlight contains UV light, UV radiation. Sunlight is a white light. Apart from it, they also contains UV radiation, UVA, UVB. So, UVA or UVB, generally, ultraviolet rays are non-ionizable in nature, which means they can't ionize actually any molecule. But if that falls into our skin or cells, after a prolonged period of time, they can cause certain changes in our DNA. That is very important, which means important in our case of mutation study, because a prolonged UV light radiation affects the change in the DNA, which can cause the change in the genome or which can lead to mutation. So it may cause diseases in us. Example, UV light can cause pyrimidine dimers, especially thymine dimers, which means the UV radiation have the enough energy to break the pi bond in between these the nucleotides. I will say it once more. UV light have enough energy to break the pi bond. We know that in a chemical structure there are two bonds mainly in a covalent molecule. I am talking about covalent molecule because they are involved in covalent bonding here, DNA mainly. So if we are hit with UV radiation for a prolonged time, UV radiations are non-ionizable in nature. They, after a prolonged period of time, they have the enough energy to break the pi bond in our DNA. So, if there is a breakage, there will be mutation forming pyrimidine dimers. We will discuss it later in the next chapter. Now, just understand like this. So, there is a mutation which can cause certain diseases in our body, which may also lead to cancer. Our DNA replication mechanism can't work properly because of a problem in the DNA. So this is the condition. It is caused by the non-ionizing radiation. You look, even the UV light prolonged exposure can lead to disease. So it is better you stay away from sunlight by either blocking the sunlight by using physical methods like using an umbrella going outside or covering your body, or you can use sunscreen, right? Okay, I prefer you to use the first method, physical method, because the rest is the chemical, right? Prolonged usage of the sunscreen can also lead to the sunscreen gets absorbed into your bloodstream. It will also lead to some problems, right? So it is the better thing you avoid chemicals and use the physical method, right? Using any covering, cover your body while going. There is no issue. You at least you can get 10 minutes of sunlight, but I am saying prolonged. Next we are talking about the mutating agent which are ionizable in nature. Ionizable in nature, which are very serious issue. They can ionize the molecules. Some of the examples are X-rays, alpha, gamma rays, etc. These waves have the enough potential energy to break the sigma bond of your DNA. That can even lead to more diseased conditions. So this is actually mutation. I think you have understood it very clearly. Okay. Now we are going to the next part, which is the genetic disorders. A very easy topic, genetic disorders. In this topic, you need just to understand what is the disease. What is the disease condition? Okay, you need to understand the symptom of the disease and the disease condition. When will this disease occur? Bus. Then this portion will be easy for you. So, genetic disorders can be classified into Mendelian disorders and also chromosomal disorders or chromosomal aberration or chromosomal abnormalities. So I will write it here. Genetic disorders can be classified as Mendelian disorders. Mendelian disorders and also chromosomal abnormalities. This is the case. Chromosomal abnormalities, or it is known as chromosomal aberrations. Okay, because of the change in the chromosome. So first I think we will discuss about chromosomal disorders. Okay, chromosomal disorders is due to the change in chromosomal disorders can be divided into two. What is chromosomal disorders? Chromosomal disorders is actually the either the increment or decrement in the chromosomal number, chromosomal gene number. Okay, I will say that chromosomal aberration is the insertion or the duplication of a segment of a DNA which result in the alternation of the chromosome. Okay, so chromosomal disorder, what are actually chromosomal disorder? Chromosomal disorders are actually caused due to the sometimes the addition or deletion of the certain segments of the chromosome. Addition or deletion of the chromosome. Addition. This is deletion. Consider this has been deleted. This has been removed. This portion has been removed, or a certain portion has been added into the chromosome. You will clearly understand how it is added or deleted in the next chapter. Now just understand like this. So chromosomal disorders are either the insertion or the deletion, or we can say insertion means duplication also. So chromosomal disorders can be divided into two. First is the structural chromosomal aberration and also numerical chromosomal aberration. First is the numerical chromosomal and also second is the structural chromosomal. What is structural chromosomal and what is numerical chromosomal? First we will discuss numerical chromosome. What is numerical chromosomal disorders? Numerical chromosomal disorder means the change in the number of the chromosome which can lead to the alternation, which can lead to the chromosome disorder, which means actually the change in the number of chromosome means change in the number of DNA. Okay, how this change in the number of DNA is caused by certain mutation or by the internal activity of the cell. So numerical chromosomal aberration means the change in the number of the chromosome. We are in numerical, we only talk about the number. Okay. In case of the structural chromosomal aberration, we will only talk about the structure of the this portion, structure of the DNA, structure of the chromosome. In numerical, we will only talk about the number in the DNA, and we will in structural we only talk with the structure. But in your textbook, I will show your textbook. Look, color blindness. Look. Look, in your textbook, it is only mentioning about ploidy, which means the number, chromosomal number aberration. It is not talking about structural aberration. So we will be only discussing about your numerical aberration or numerical chromosomal aberration or numerical chromosomal abnormalities. What is structural? Structure means change in the structure, either the deletion of the either the insertion of a new structure or the deletion of the structure in the chromosomal disorders. Like example is the transversion, inversion. These are the examples of the structural chromosomal aberration. Okay. Now we will be dealing about numerical chromosomal aberration. So in numerical chromosomal abnormalities, we are talking in the change in the number of chromosome. Change in the number of chromosome. It is either the changes, the addition of a chromosome, or the removal of the chromosome in the cell. Okay, we will understand it. In numerical chromosomal aberration abnormalities, we are talking about the change in the chromosome number. It is not the chromosome structure. We are not dealing about chromosome structure now. We are dealing about the chromosome number. It is by the addition of a chromosome or a deletion of a chromosome. So, and it is not a complete set. If it is a complete set, we will divide it as polyploidy, and if it is only a particular number of chromosomes, we can divide it as aneuploidy. So, we can divide the numerical chromosomal abnormalities into aneuploidy, and polyploidy. Polyploidy or euploidy. So, chromosomal numerical abnormalities can be divided into aneuploidy and euploidy. So, what is aneuploidy? Aneuploidy is the change in the number of chromosome either by the addition or the deletion of less than a complete set. So, you can write the definition as, what is aneuploidy? Aneuploidy is the change in the chromosome number. It is by either the addition or the deletion of a less than a chromosome set or a less than a set of chromosome. So what is polyploidy or euploidy? Polyploidy is the entire deletion or addition of an entire chromosomal set. So I think you have written the definition. Now we can go into the structure. How this definition is, how we can discuss what is the definition by drawing a diagram. Look. So consider a cell which is diploid in nature, and the diploid condition 2n is equal to 6. Okay, a cell is having a chromosome, 3 set of chromosome. Okay, 3 set of chromosome, total 6 chromosome, individual chromosome. So I am writing the chromosome here as chromosome 1, its homologous chromosome, chromosome 2, its homologous chromosome, and chromosome 3. Okay, look here. After cell division, which means anaphase, after cell division, 2 cells are formed. Okay, 2 cells are formed after the cell division. So in one cell we just found that this is the condition 1, 2, 2, 3, 3. And in the other condition, in the other cell, we just found this. So you just look after cell division, just consider a condition, after the cell division, after mitosis, just consider after a mitosis, the cell was formed, 2 cells were formed, which we know that it is diploid in condition. In one cell, there is an extra addition of a chromosome here, and the other cell, there is a deletion of a chromosome. One chromosome pair is missing, a homologous chromosomal pair is missing. Okay, this is known as numerical abnormalities, numerical chromosomal abnormalities in a cell. So in this condition, we can say that this is hyperaneuploidy. What is it? Hyperaneuploidy. Okay, I think you understand it. This is known as hyperaneuploidy. And this is known as hypoaneuploidy. Why it is known as hypoaneuploidy? Because it is having a chromosome which is less than that of the normal chromosomal number. So we can divide aneuploidy as hyperaneuploidy and hypoaneuploidy. Okay. So further, hyperaneuploidy, which means the increment in the chromosomal number. Some of the examples of the hyperaneuploidy are trisomy, double trisomy, and tetrasomy. You can write it down. Some of the examples of the hyperaneuploidy are trisomy, double trisomy, and tetrasomy. We will discuss it right now. What is this? And in case of hypoaneuploidy, means there is a decrement, there is a deletion, or one chromosome is missing, one homologous chromosome is missing. This is known as hypoaneuploidy. I first said what is aneuploidy. Aneuploidy is the change in the number of chromosome by the deletion or addition of a chromosome which is less than that of a complete set, not a complete set. If it is a complete set, then it is the definition for polyploidy. We will discuss how an entire set is removed. Why this occur? Why there is an increment in the hypo and why there is missing? Because it occur in the anaphase phase. Anaphase usually, because we know that in anaphase the chromosome, both the homologous chromosome get separated, right? Or if it is in case of meiosis 1, it is the separation of the homologous pair of homologous chromosome. But if it is in case of anaphase 2, then it is the pairing of, then it is the separation of the sister chromatids. I have mentioned it in the previous lectures, how this separation occurs. You can, if you know the cell division, you will understand it very clearly. So while the separation, if there is an issue in the separation process, then this condition will occur. This is the case I said in the internal problem. If the cell faces the internal problem, this condition can occur. Non-disjunction. It is known as non-disjunction in the anaphase phase. If the segregation or the separation is not proper, then this condition may arise. One chromosome may stick with the other, thus forming a extra chromosomal pair. Okay, I think you understand it. So aneuploidy can be divided as hyperaneuploidy and hypoaneuploidy. Some of the examples of hyperaneuploidy are trisomy, double trisomy, and tetrasomy. Some of the examples of hypoaneuploidy are monosomy, double monosomy, and nullisomy. Okay, in your textbook, I'm going to your textbook right now. In your textbook, look, 3 diseases, 3 condition syndrome have been explained. First is the Down syndrome, next is the Klinefelter syndrome and Turner syndrome. Usually 3 are given in your textbook. So these are the examples of hyperaneuploidy which comes under the trisomy. So we are going to discuss what is trisomy. Okay. So we are going to discuss trisomy. Please write it down. Trisomy. How trisomy occur? Okay. Trisomy is the example of hyperaneuploidy, which means increment of a chromosome number. So trisomy, double trisomy, and tetrasomy. First we are going to discuss trisomy. What is the condition of trisomy? The condition of trisomy is if the organism is 2n in condition, then we know that 2n plus 1. 2n plus 1 is the condition of 2n plus 1 is the condition of trisomy. What does it mean? It means it have a 2n, the organism is normal 2n, but it have an extra chromosome of 1. 1 means it has an extra chromosome, which is in your, this, look, this is the condition. This is an example of trisomy, which is having an extra chromosome 1. We should understand that either 1 can come, either 2 can come, either 3 can come. It is according to chance. I have just written 1 because I have just written as it is. Okay. The condition may, it can also occur, but here 3 will not be there. Okay. This is the condition of trisomy. Okay. So the example of the trisomy in your textbook are is given as Klinefelter syndrome, Down syndrome, and also certain other syndromes are also there. Patau syndrome, you know Patau syndrome, P-A-T-A-U, the Patau syndrome, Down syndrome, Klinefelter syndrome. These are the examples of trisomy. So, in case of Down syndrome, first we are going to discuss about Down syndrome. There is another example also, Edward syndrome. This is also an example of trisomy. I will write the examples here. First, we are going to discuss about Down syndrome. Down syndrome. So, in Down syndrome, I will write the Down syndrome condition. 47, comma, plus 21. Next, Klinefelter's syndrome. Klinefelter's syndrome. What is the condition for Klinefelter syndrome? 47, comma, X, X, and Y. Next, Edward syndrome. Edward syndrome. (47), 18. My handwriting is bad because of this system. Sorry for that. Next is Patau syndrome. It is having 47 and plus 13. Okay. This is the 4 examples of the trisomy. First is the Patau syndrome. Just consider this in it is in order 18 plus 18. Next, Down syndrome plus 21. Sorry, Patau syndrome plus 13, Edward syndrome plus 18, Down syndrome plus 21, and Klinefelter syndrome XXY. So just consider this in order 13, 18, 21, XXY. So these are the examples of the trisomy. What does it means? It means that in Down syndrome, we know that Down syndrome is usually seen in human beings. It is a syndrome. Usually we are having diploid in nature, and our diploid condition is 46. If an organism, if a human being with a Down syndrome will have 47 chromosome. How 47 chromosome? Because of the increment in one chromosome, any of the one chromosome. But the increment here is in the 21st chromosome. We have 23 set of chromosome. In Down syndrome, the 21st chromosome will have an extra 21 copy. This is the 21st chromosome. This is the condition. An extra 21 copy of 21st chromosome will be present in a human being with a Down syndrome. So the total chromosome number will be 47. Odd. It became odd. Earlier it was even in number because of the pair. Now an extra chromosome in the 21st, so it become 47. This is the same case in case of Edward syndrome and Patau syndrome, which is having 13, 18, 21 increment. In Klinefelter syndrome, the increment is in the X. One more X is being added. Okay. So there it became 47 total number. So we will discuss about the disease. Look, first in your textbook, the discussion is about the Down syndrome. Down syndrome, I said 21 copy. It is a trisomy, example of trisomy, 21 copy. Okay, what is the symptom? An organism the human being will have a small round head with a furrowed tongue and partially open mouth. So, and the palm is broad with characteristic palm crease. Physical and psychomotor and mental development is retarded because actually of the presence of an extra copy in the 21st chromosome. These are the conditions for the Down syndrome. And what is the genetic number? 47 and 21, right? So this is the case of Down syndrome. In Klinefelter syndrome, we know that 47 total number because it is an example of trisomy, and extra copy of X is found in the individual. So such an individual will have an overall masculine development. However, the feminine development is also expressed because usually in XY it is a male. So an extra X is being added. So the organism will have, the human being will have a feminine in nature, and the individuals will be sterile. They can't reproduce. Okay. Next, the example is about the Turner syndrome. Turner syndrome is it is absence of one X chromosome, thus leading to 45 with XO. So okay, now we have, okay. This is an example of hypoaneuploidy. So we have discussed there is also one more disease, which is the Turner syndrome, which is an example of hypoaneuploidy. What is hypoaneuploidy? Hypoaneuploidy means there is a absence of a chromosome. There is a decrement in the chromosome number because of the deletion of a chromosome. So hypoaneuploidy can be divided into monosomy and monosomy, double monosomy, and nullisomy. Trisomy can be divided as hyperaneuploidy can be divided into trisomy, double trisomy, and tetrasomy. Actually, there is no need to study it in much detail, but even though I will say in double trisomy, the condition will be 2n plus 1 plus 1, which means this is the condition. Which means there is an increment in the two chromosomes which are not equal. You understand? Either increment or 1 or 2, 2 or 3, or 3 or 1. Okay. So, but the chromosomes are not same. This is an example of double trisomy. So 2n plus 1 plus 1. So the organism will be having 48 chromosomes. Next is the tetrasomy. Tetrasomy, like it is tetrasomy is 2n plus 2, which means the two chromosomes have been increased and the two chromosomes are either same. The two chromosomes are same, which means 1 can come, or 2 can come, or 3 can come, which means two chromosomal increment. This is the case of double tetrasome. Now we are going to talk about hypoaneuploidy. Hypoaneuploidy means it is the decrement in the chromosome number. So hypoaneuploidy can be divided into what? Hypoaneuploidy can be divided into monosomy, double monosomy, and nullisomy. So Turner syndrome is an example of monosomy. What is monosomy? Monosomy means 2n minus 1. 2n minus 1 is the example, or 2n minus 1 is the case in nullisomy. So Turner syndrome is an example of nullisomy. Sorry, not nullisomy, monosomy. Monosomy means 2n minus 1. Double monosomy means 2n minus 1 and minus 1, which means the two minus decreased chromosome are different. And in case of nullisomy, that 2n minus 2. So hypoaneuploidy can be divided as monosomy, double monosomy, and nullisomy. In monosomy, decrement of one chromosome, any one chromosome, and in double monosomy means two different chromosome decrement. Either 1 and also 3. This is the condition. In nullisomy, two same chromosome have been decreased. Two same chromosome have been decreased. So in case of monosomy, the example is Turner syndrome. So what is the condition of Turner syndrome? So 46 minus 1. So 45 chromosome. 45 chromosome. So the missing of one X chromosome. XO. XO means there is no chromosome there. So this is the case of Turner syndrome. So Turner syndrome, XX, 1X is removed. So XX means it is seen in females. The organism or the human being will lack secondary sexual characters, as that of female secondary sexual characters will be lost. This is the case of Turner syndrome. Actually, how you need to study is that you need to study the condition for the occurrence of this syndrome, and also the symptoms of this syndrome. This much is required for the NEET purpose. Next, polyploidy. What is polyploidy means? Polyploidy is the change in the increment or the decrement of the entire set of the chromosome, which means in polyploidy this entire one set will be decreased, making it in a haploiding condition. So entire set will be decreased or an entire new set will be added. So this is the condition of polyploidy. Next, we are going to discuss about chromosomal disorders in so now we have discussed about the chromosomal abnormalities. Now the next one is we are going to discuss about the Mendelian disorders. So in Mendelian disorder, why it is given as Mendelian disorders? Because these disorders have been passed to the next generation according to Mendelian principle of inheritance. The disorders have a exact passage to the next generation by following Mendelian principle of inheritance. So in order to understand the passing over of this disease to the next generation, we need to understand how it is passed. So first of all, we need to understand pedigree analysis. So there are certain shapes and structures we need to understand in pedigree analysis. What is pedigree analysis? So it is the the disorders are transmitted to the offsprings on the same line as we have studied in the principle of inheritance. So this is so we can easily understand it by studying pedigree analysis. Pedigree analysis can easily from pedigree analysis one can easily understand the trait in question, whether it is dominant or recessive. The pattern of inheritance of Mendelian disorders can be traced in a family by the pedigree analysis. Okay. So there are certain shapes and structures for pedigree analysis. Now we are going to write it. So in pedigree, actually from here I will say the order how we will be studying. First we will be discussing about the Mendelian disorders. After that we will go to pedigree analysis, and after that we will discuss each and every disease in detail. Okay. So in Mendelian disorders, the Mendelian disorders follow the Mendelian pattern of inheritance. Okay. So we can classify, or I have grouped the disorders which is given in your text according with their general definition. Okay. With their general heading. So we can divide it into actually 4 types. First is the X-linked recessive, second X-linked dominant, and autosomal dominant and autosomal recessive. So I have classified the diseases into 4 main groups. First is the X-linked recessive, X-linked dominant, and autosomal recessive. So first we are going to deal with the X-linked recessive. Okay. So I will be writing XR. You should write it as X-linked recessive. So in X-linked recessive condition, X-linked recessive and X-linked dominant. So we are here talking about the sex-linked mutation. What happens in a sex-linked disease condition? So we are dealing with the condition when the mutation is occurring in the sex chromosome. So here we know that human beings have two types of sex chromosome, X chromosome and Y chromosome. So here we are dealing with the sex linked X recessive diseases. Okay, so what is X linked recessive? We know that in male it is XY and in case of female it is XX. So in X linked recessive the condition is like this. So we know that female XX, male XY. So this condition occur, which means the sex linked recessive disorders or mutation occur when the X is in its recessive condition. The disease only occur when it is affected in both the chromosome, both the X chromosome. I think you are understanding it. This disease occur only when the disease in both the X chromosome. If both the X chromosome are mutated, I have written put a dot for mutation. If both the X chromosome are mutated, then it will show the diseased condition. It will show the diseased condition. When there is only one mutation, there is a mutation in only one X, then it will show a normal progeny. It will be normal, but it is also a carrier of the disease. We will understand how it is working as a carrier. In case of male, usually males are having only one X chromosome. So all the males with X mutated will be diseased in condition. This is the case of X-linked dominant, X-linked recessive condition. So this is the case of X-linked recessive condition. The examples of X-linked recessive diseases are hemophilia, okay, color blindness, Dutch muscular dystrophy, Hunter syndrome. Actually, out of these four, usually these are the four common we have studied, but in your textbook, hemophilia and color blindness have been given. Actually, we will study it in very deeply after we discuss this classification. So, what I have said is that in X-linked recessive, we can only say the disease will occur only when both the X are mutated, both the X chromosome are mutated. The disease occur only when both the X chromosome are mutated. Okay, and in case of this is in case of female. In case of a male, there is only one X chromosome. So if the X is mutated, then we can say that the male will be diseased condition. But in case of female, if one X is affected, out of 2X, it is in a heterozygous condition, right? So if one X is affected, the progeny or the organism will be normal, but it will be a carrier for the next generation. We will discuss how it is being carried out. Okay, so this is the case of X-linked recessive disease. So next we are going to discuss about X-linked dominant. Okay, next is about the X-linked dominant. I will be writing XD, X-linked dominant. So what is the condition for X-linked dominant? In X-linked dominant, I will write XX, XY, this is male, this is female. So the disease occur when it is in the disease when the mutation is in the dominant condition, which means even if one X is present, the organism will be diseased. We know that this is already diseased because of only one X in male. So what is the condition if two are present? Two are present, it will be sure, it will be diseased condition. Okay, this also will be diseased condition. This is the case of X-linked dominant. Usually you should understand mutation in its dominant form are very rare. Mutation in the dominant form are very rare. Mutation can occur even though it is rare in recessive disease. Mutation can occur in recessive condition mainly because the other chromosome can mask the effect, mask this mutation, because we know that if the organism or if the genotype is in the heterozygous form, the dominant allele can mask. So usually the mutation in the dominant allele are very rare. Okay, so this condition is very rare, mutation in the dominant allele, this condition, and this is very rare because two dominant form has been mutated. Okay, this is the condition in X-linked disease. So this is the case of X-linked dominant. Now we are going to discuss about autosomal recessive disease. Autosomal recessive. Autosomal recessive, AR. Okay. What is autosomal recessive? In autosomal recessive, the mutation is in the autosomal genes. In case of sex-linked mutation, which is X-linked recessive, X-linked dominant, and also there is also one more thing, Y-linked gene, Y-linked disease. Actually, there is no need to study much because I have been classified it into group. We are studying this much. Okay, so in case sex-linked traits, sex-linked traits are X-linked recessive and X-linked dominant. In case of autosomal mutation, in case of autosomal mutation, we can divide it into autosomal recessive and autosomal dominant. Okay. So, what is the condition in autosomal dominant, autosomal recessive? I am taking capital A, capital A, capital A, sorry. I am taking capital A, capital A, capital A, small a, and small a. The disease occur only when the autosome are in its recessive condition. In this condition only the disease can occur. This progeny will be normal and this progeny also will be normal. Okay, this is the condition in autosomal recessive. So the condition only occur when the autosomes are in its recessive form. Some of the examples of the autosomal recessive diseases are phenylketonuria, okay, xeroderma pigmentation, albinism, cystic fibrosis, thalassemia, which is given in your textbook. Only autosomal recessive disease which is given in your textbook. These are the some examples. Sickle cell anemia. These are some examples of the autosomal recessive disease. So I will say it once more. The examples of the autosomal recessive diseases are sickle cell anemia, thalassemia, albinism, xeroderma pigmentation, cystic fibrosis, also phenylketonuria. These are some of the examples in your textbook. Phenylketonuria is mentioned, thalassemia is mentioned, sickle cell anemia is also mentioned. Okay, this is the case of autosomal recessive disease. So while we are analyzing the pedigree, if we are doing questions in pedigree, you will understand it more deeply. Okay, so this is the case of autosomal recessive. Now the next thing is the autosomal dominant. What is the condition in autosomal dominant? The in autosomal dominance, we should understand that AA, capital A, small a, and small a, small. In autosomal dominant, the disease condition occur in dominant form. So this is diseased. So this is also diseased, and this is a normal project. Okay, some of the examples of autosomal dominant disease are Marfan syndrome, achondroplasia, polydactyly, brachydactyly, etc. Okay, these are the examples of autosomal dominant diseases in your text. Actually, it is not mentioned in your textbook. I have just said it. And also neurofibromatosis. I am saying this because if you are going through the questions, sometimes you will get to know about the disease, the names. Okay, so this is the condition of the Mendelian disorders. So actually, Mendelian disorders transmission or the disorder pattern, the disease inheritance pattern is same as that of Mendelian inheritance. So we can classify it into 4 types actually. First is the X-linked recessive, and X-linked dominant, and also Y-linked. These 3 are actually linked to the sex chromosome. And the next is the autosomal dominant and autosomal recessive. These diseases are actually transmitted through the autosomes. Autosomes are the chromosomes other than that of the sex chromosome. Okay, so this is the pattern of transmission. I have said the examples. In X-linked recessive, some of the examples are color blindness, hemophilia, Dutch muscular dystrophy, and Hunter syndrome. X-linked dominant, there is actually no disease mentioned in your textbook. And in autosomal recessive, Some of the autosomal recessive diseases are phenylketonuria, albinism, xeroderma pigmentation, sickle cell anemia, thalassemia, etc. Okay, actually in your textbook thalassemia, sickle cell anemia and phenylketonuria are discussed more deeply, and cystic fibrosis also. In case of autosomal dominant disease, some of the autosomal dominant diseases are Marfan syndrome, achondroplasia, Huntington disease, polydactyly, brachydactyly, nail patella syndrome. Okay, these are actually the diseases which are mentioned. Actually, this is not mentioned in your textbook. I'm just saying for your information. So these are the general 5 classification. There is also one classification, one group which is Y-linked group, which is transmitted from the grandfather to the father to the next progeny. So these are actually the transmitting pattern. We will do it very clearly while we are analyzing the pedigree. So if we are doing the pedigree questions, we can do it very clearly. Actually, I have just set it on the basics. Okay. This is actually the things to be delivered. Next we are going to study the each and every diseases in detail. So first disease we will go through in an order. First we will be dealing with color blindness and also hemophilia. Okay. I think you have noted down the examples of each and every groups. So first we are going to look about color blindness. I have taken some pictures of color blindness. I think you have written it, right? So first we are going to discuss about the color blindness. What happens in color blindness? Actually, from the word color blindness, we can understand that the organism, the human beings who is suffering from this disease can't see certain colors. He or she is blind for the color. Yes, he or she is blind for the color, and we know that in a person with color blindness is having an X-linked recessive trait. He follows a X-linked recessive trait in which if he is a man, he's a male, he will be having this condition. He will be having X, Y, and this will be the condition of a male, and the condition of a female will be like this. What happens a female with this genotype? She is not colorblind, but she is a carrier, because in the next generation she can transfer her disease. Okay. So actually this concept is very much to be set in deep. I think so. If there is need for it, we will be discussing it while we are doing the questions. So in case of a color blindness, we know that actually color is perceived from our eyes, right? We can understand or we can sense it by using our organ, which is the eye. Eye, in its retina portion, we know that I have a retina. Inside the retina, there is the presence of certain cells, certain light perceiving cells or light receiving cells. So these cells is known as cone cells. Cone cells actually can absorb the light and send the signal into the brain and say that this is the light which I am seeing. Okay. Actually, the cone cells are actually the protein molecules, right? So there are actually 3 protein molecules in the cone cells which have been identified for actually 3 color, which is for red, blue and green. Sorry, for 3 color, red, green and blue. So there will be 3 proteins for this perception. 1 protein for green, 1 protein for blue and 1 protein for red. We need to understand from these 3 protein, only 2 protein are seen in the sex chromosome, which is our study, right? We are dealing with sex chromosome, X and Y, and color blindness is a sex-linked trait, which means X-linked recessive. And so in the 3 proteins, only 2 proteins are present in the X chromosome, and the other protein which receives the blue light is present in the autosome. Actually, we need to consider only the two proteins which are sex linked, which are green and also red in color. So a person with color blindness have a mutated protein which can't absorb the two lights, which are the red light and the green light, because the corn cells lack the pigment which absorbs these two lights. This is the condition for color blindness in human beings. So what happens? Color blindness is actually not a serious issue in our society because only two of the color are missing, right? So what happens in a color blinded organism or a human beings? He can't see the two color which is actually so red and green, right? He can't see, or she can't see the red color, and she can't see also green color. For them, actually, it will be an intermediate color. So they can't identify actually which color is this. So for if you are having color blindness, you can check it right now. If you are having color blindness, you will not be able to see, look in this diagram, you will not be able to see what shape and what color have been encoded here. You will not be able to see the color inside and the shape inside. Actually, we know that it is those who are not having color blindness can understand this first diagram is having a shape of 12, number 12, in which it is coded in red color, actually red green color. Okay, so the person with color blindness can't see what is inside. For them, it is actually the mixture of color, right? He can't be able to, or she can't be able to identify what's inside. So this is the case of color blindness. So color blindness is a X-linked recessive disease. Okay, I think you have understood it. The next disease to be discussed is the hemophilia. I think this much is required for color blindness. There is nothing much to be seen. Next is the hemophilia. We will go to your textbook. Hemophilia, right? Hemophilia is also a X-linked recessive disease. I have mentioned it. So how is hemophilia caused? Hemophilia in a organism, in a human being with the hemophilia, a protein which is used in the cascade of protein involving the blood clotting is absent. So the blood won't clot. So if there is a cut in a human being or organism, he will be continuously bleeding. This is the condition of hemophilia. Okay, next we are going towards the other three diseases, which are sickle cell anemia, phenylketonuria, and thalassemia. A very easy topic, right? So what is sickle cell anemia and which category does it belongs to? Right, we know that sickle cell anemia belongs to the autosomal recessive disease, right? It is a autosomal based inheritance pattern. So what some of the examples of autosomal recessive diseases are sickle cell anemia, phenylketonuria, thalassemia, albinism, xeroderma pigmentation, cystic fibrosis, etc. Okay, these are the examples. So what happens in a sickle cell anemia? We should understand that sickle cell anemia, there are two words given in it. First is sickle cell. What is sickle? We know that sickle is a tool, right? I think we have seen it. Sickle is a tool which we use for cutting grass, some other things, right? Which is used in the agricultural purpose. So sickle shape anemia. Anemia means low level of red blood cells in your body. So sickle cell anemia, what happens in sickle cell anemia? It is a autosomal recessive disease. I will say story, you should just understand it. There is nothing much to study, just understand the symptom, just understand the condition. What happens in a sickle cell anemia? Sickle cell, we know that in human beings, our oxygen is transported throughout the body with the help of red blood cells, right? Red blood cells are the particular cells which are used for the transport of oxygen throughout our body. How it is transported? To understand this, we need to understand the structure of RBC. RBC is a cell which lacks nucleus. We know that most of the cells in our body or cell contains nucleus, right? In RBC, the nucleus is absent. So we are going to deal with the structure of RBC. Structure of RBC. Actually, I have given it here. Structure of RBC. red blood cells. Red blood cells contain hemoglobin. Red blood cells contain hemoglobin. From the word hemoglobin, there's two words, right? Heme plus globin. So hemoglobin consists of a heme part. Heme means iron. I-R-O-N, iron. And also globin, which is a protein part. So hemoglobin consists of iron part and also a protein part. First, we are going to discuss about the iron part. Sorry, the globin part, which is a protein. We know that protein consists of amino acids. Proteins are made up of amino acids. Like amino acids join together to form a large protein. So many large protein aggregate to form a very large protein. Now we can understand like this. Okay. So in hemoglobin, it contains a heme part, which is the iron part, and also globin part, which is the protein part. This is the protein part, the globin part. Okay. So the globin part actually is a tetramer. Tetramer means 4 units. It contains 4 units. 2 alpha and 2 beta. So we can write it as alpha 2, beta 2. Alpha 2 and beta 2. This is the protein part of the hemoglobin, alpha 2, beta 2. So each, which means it contains 2 alpha, 2 beta, thus forming a tetramer, right? Each subunit means each alpha, each beta contains other part, which is a heme part, right? Which contains a heme part. Here, heme means iron. Iron is attached in a porphyrin ring. Just understand this. There is nothing to study. Just understand how it is, because we are just studying the condition of the sickle cell anemia. For that, we need to understand the structure. So, what happens? So, each unit have a porphyrin ring which contains iron. Okay, which is the heme part. So, 4 heme parts. Each heme parts can accumulate oxygen molecule. So 4 oxygen molecules total they can accumulate 4 oxygen molecules each. So that is the condition. So there is 4 heme in 4 porphyrin ring. Okay. So there is 4 iron, and each iron can take each oxygen. So oxygen molecule. So there are 4 oxygen molecules, and a molecule and a hemoglobin can actually take 4 oxygen. A hemoglobin can take 4 oxygen molecules transport. How it is taking the oxygen molecules? Because it is having the presence of Fe2+. So 1 iron can take 1 oxygen molecule. Molecule means O2. So 4 iron can take 4 oxygen molecule. 4 heme can take 4 oxygen molecules. So what happens in sickle cell anemia? Actually, this is the structure of the hemoglobin. So what happens in a sickle cell anemia? In sickle cell anemia, the mutation is autosomal recessive. Autosomal recessive means the mutation, the disease can only occur when the genotype is small a. So in sickle cell anemia, the beta chain, I have said that beta chain, there are 2 beta chain. One beta chain has 141 amino acid, and the other beta chain has 146 amino acid. I have said that beta, alpha, everything is protein. Protein is made up of amino acid. In beta, one beta contains 141 amino acids, and the other beta contains 146 amino acids. So in sickle cell in a sickle cell anemia, alpha contains 141 amino acid and beta contains 146 amino acid. So in a human being with sickle cell anemia, the sixth position of the amino acid, which is glutamic acid, is mutated to form other amino acid, which is valine. Okay, what happens? In the beta position, 6th amino acid, which is normally means in a normal person, it is actually glutamic acid or glutamic acid, which is a acidic amino acid. This acidic amino acid gets changed into valine, which is a hydrophobic amino acid. Actually, amino acids are certain groups. Just understand, 6th position of the glutamic acid, which is acidic, just changed into a hydrophobic amino acid. So how it affects the shape of the cell? Due to this mutation, the hydrophobic valine interact with the other hydrophobic amino acids near. There is so near amino acids. I said it is having a 146 amino acid, beta, alpha, it is 141 amino acids, and thus a mutated amino acid, which is the valley, interact with the other amino acid, and thus they form a hydrophobic chain and they clump together. And they will form a sickle shaped structure inside the RBC. So the cell will also become sickle shaped. Sickle means this is the condition. Concave, it became a biconcave in structure. Biconcave is actually like this, sickle shape. So this is actually the condition. Actually there is nothing to be studied, but just understand the concept. So what happens? This sickle shaped RBC, while flowing through our blood vessels, actually blood vessels are very small that they can only transmit one RBC at a time. So a sickle celled RBC, while transporting into the through the blood vessel, it is very difficult for them to transport, and what happens? They break due to the small size of the blood vessels. The RBC gets break and destroyed. So anemia condition occurs, anemic condition, which means low level of RBC, which in turn will contain low level of oxygen, because RBC will take one hemoglobin can take 4 oxygen molecules. So here hemoglobin is destroyed, RB is destroyed, so the person will suffer from anemia. This is the condition of sickle cell anemia. Next, we are going to deal about the next disease is the hemophilia we have described. Next is the thalassemia. Thalassemia is autosomal recessive, as same as sickle cell anemia. What happens in thalassemia? I think you have understood the condition, how thalassemia occurs. Now we are going to say what is the symptom, what is the condition, what occur in thalassemia. We know that thalassemia can be divided into alpha thalassemia and beta thalassemia. What happens in alpha thalassemia? Alpha thalassemia means there is the destruction in alpha chain. In beta thalassemia, beta chain. Thus, it will in turn destroy the RBC. So, in turn, the person will suffer from anemia, which means lower level of RBC. This is the condition. Okay. Now, we are going to discuss about phenylketonuria. Phenylketonuria. Okay. Phenylketonuria. Phenylketonuria is also an example of autosomal recessive trait. What happens in a phenylketonuria? We know that phenylalanine is an amino acid. Actually, phenylalanine needs to be converted into tyrosine with the help of PAH gene. In a person with phenylketonuria, the PAH gene is mutated because of the condition, autosomal recessive condition, small a. So the PAH gene can't be synthesized, can't be produced. Gene will not be expressed. So phenylalanine gets accumulated. So phenylalanine can't be converted into tyrosine. Tyrosine is a molecule which can lead to two other pathways. First is the melanin synthesis. Second is the certain central nervous system receptor molecule production, like catecholamines. Okay, actually there is nothing to be studied, just understand. So a person with phenylketonuria can have central nervous system issue, and also he will be having no production of melanin, which means he will be having white colored hair, etc. This is the condition. So the accumulated phenylalanine can be converted into phenylpyruvic acid, which is toxic to the central nervous system. So phenylalanine needs to be converted into tyrosine with the help of PAH gene. Here PAH means phenylalanine hydroxylase enzyme. This enzyme is absent because of the mutation in recessive autosomal. So this is the condition of phenylketonuria. Actually, we have discussed all the Mendelian disorders and also chromosomal disorders. Now there is a small topic which is pedigree analysis. Pedigree analysis will be discussed while we are doing the questions. Actually, there is nothing to be discussed in pedigree analysis. We can only understand while we are doing the questions. And though I will say some basics of pedigree analysis. In pedigree analysis, we give certain symbols for the analysis of the pedigree, which means analysis of the family trait. In pedigree analysis, we give a square for male, a circle for female, and if the male is affected, we will just draw lines. Okay, and if the female is affected, this is the condition. If the male is the carrier, we will draw a dot here. If female is the carrier, we will draw a dot here. If there is a mating between the male and female, we will be drawing like this. And if they are having a progeny, we will be drawing like this. This is the progeny, which means the children of these male and female, father and mother. This is the first generation. This is the second generation. These are usually drawn in Roman letters. And the offsprings are marked according to number 1, 2, 3. First produced, second produced, third produced, fourth produced. These are their parents. So this is actually the condition of pedigree analysis. Pedigree analysis can be fully understood while we are doing the questions. Actually, this is the end of your lecture, end of this chapter. Now in the next coming lecture, we will be discussing each and every questions very deeply. I think you have understood this lecture. If you are having any sort of doubt, you can comment me in the comment box. Okay, thank you.